All right, so welcome everybody. I'm really excited to have Dr. Beth Cimini from Broad Institute for our week 6 of our invited speaker talk. She is going to talk about making most of the microscopy data with high content image analysis or in general analysis. Again, on behalf of Team ICI, I'm really excited to have her. Dr. Cimini is a senior group leader at Broad Institute. She has her own group. She is also a Chan Zuckerberg Institute imaging scientist. She is one of the person who has been part of the CellProfiler team for a long time now. She is also part of the Center for Open BioImage Analysis, along with a few other people who are part of this center. Before getting into all of this, she has traveled both the ends of the country, both the East and the West Coast. She did her undergrad research in visual neuroscience from Boston University. She obtained her PhD in biochemistry and molecular biology with the famous Dr. Elizabeth Blackburn at UCSF. One of the things that was common at both the places is that her projects were related to image analysis. And following this, she joined Dr. Ann Carpenter's group at Broad Institute, where she led a team collaborating with more than 30 outside scientists per year with custom bioimage analysis projects. And that's where CellProfiler really comes into picture. At the moment, Dr. Cimini's lab focuses on bioimage analysis tool creation, for example, image annotation tools such as Pixme, as well as maintenance of the CellProfiler platform as a whole, as well as applying new tools to address novel biological problems. More importantly, she has also created a postdoctoral training program in bioimage analysis, and she also heads and leads the Broad's effort towards community engagement driven processes for biological projects for COBA. So without further ado, I would like Dr. Cimini excite us with all the fun stuff she has for us. Take it away, Beth.
Yeah, thank you for the introduction, and thank you so much for having me. Wrong desktop. I want to share that one. Yeah, I'm really excited to be here and chat with everybody. My understanding is that most of you were at the description of CellProfiler last week in the sort of introductory workshop. So my talk is going to be in 2 parts. I'm first going to talk about high content analysis and doing high content analysis with CellProfiler. What are some considerations in putting together a high content analysis experiment? And in the second half of the talk, I'm going to talk about some of the things that you can do with high content analysis that are hard to do unless you are using an automated software to generate lots and lots of measured features from every image that you have. And so hopefully that will motivate why tools like CellProfiler are really valuable. I'm going to be presenting work. So my group is very new. Most of the work was done under the auspices of what is now our sister lab, the Carpenter Singh Lab, which was run by Ann Carpenter and Shantanu Singh. Hopefully by the fact that all of you are here, you're on board with the idea that images contain a tremendous amount of information and are a really valuable and valid source of quantifiable biological information. We're passionate about using images because you can get single cell resolution, they're quantitative, you can multiplex things, you can cover complex model systems. But even if you don't think that you need biological image analysis software, there's one aspect that I think gets overlooked a lot of times when people talk about image analysis, which is just that human brains are terrible at quantification. It's not what they're designed to do, and they do it actually extremely poorly. Looking at this picture sort of quickly, or if you're looking at it on a small screen, this probably looks to you like a full color picture. It's not. This is a picture that is mostly grayscale with a few colored lines drawn on top. But your brain fills in the missing details and tries to give you an idea of the whole picture, the gestalt of what's going on. That's great from an evolutionary biology, understanding a situation quickly point of view, but it means that even if you think you can tell what's going on in your images by eye, your brain might not always be giving you the whole picture. or it might be giving you a picture that's more than the sum of the parts. And so automated quantification is something that we really think is important no matter what your phenotype is. And so, like I said, I'm first going to talk about, some basic ideas of how to set up a high content analysis, particularly around measuring and scoring known phenotypes, and which is the kind of science that most people think about and most people do. And there's really only sort of three things you need to do when you're creating a good high content analysis workflow. The first is if you know that there are artifacts, things like your microscope needing to be flat field corrected, or if you know that there are things like debris, do whatever you can to get rid of those. Find the objects that you care about, and this is the hard part, and then add as many measurements as is practical. You might not agree with this bullet point yet, but hopefully by the end of the talk, I will have convinced you. In terms of finding the objects that you care about, again, that is 9/10 of the work in most high content analysis experiments. Objects for everybody are going to be different. Some people might really want to find a particular organelle. Sometimes you might want to find a whole organism. But really the sorts of principles of how do you set up your high content experiment really come down to the same thing. The idea of you want to find all of the objects you care about and only the objects you care about. And I'm not going to do a demonstration necessarily of how you do that, but I'm going to share with you the general principles that I encourage everyone in my group, and definitely also my past self who wasn't always good at these roles, on how to figure out when you should stop tuning your analysis workflow and when you should actually move on and start using it in practice. The first is just, do I generally agree with most of the object segmentations in my analysis workflow? You're not going to be getting very far unless you mostly agree with most of them. Do I have approximate, hopefully your threshold is good all over the image, but if there are cases where it's a little bit too high or a little bit too low, is that in approximately equal amounts? Likewise, if I'm finding objects, do I have approximately equal number of cases, which again is a hopefully low number, where two things that should be two separate things are stuck together, which we often call under segmentations or merges, versus a case where you have one object that is truly one object split into many, which we tend to call split or over segmentation. And does this hold across a variety of images, both negative control images or my most extreme images, whatever I think are going to be the most sets of different images from each other? Do my analysis conditions have the same bias in all of those regions? And again, we have a whole blog post that goes into more about how you might figure this out. But ultimately, if you can satisfy all of these conditions, your high content image analysis is probably good enough. And this is where people who do this professionally sort of say, all right, this is where I'm going to stop. The tool that our lab uses most of the time to do our high content analysis is CellProfiler, which was originally written by Ann Carpenter and Ray Jones. I mostly show this slide because I want to show the tremendous team of people who've been involved over the years in putting this together. The software is free and open source, and what I think makes it special is there's always been at least one biologist and one professional software engineer working together on the code base, which means that you have, the luxury of the expertise of a professional software engineer to make it very high quality. But you also have input from a biologist with microscopy training who understands how biological images work, who understands the kinds of things that biologists do and need and what we call things. And so in that sense, I think that's partly why the tool has been around for as long as it has. In software terms, it's pretty ancient. Again, I know you guys did a thing on CellProfiler last week, so I won't go into this in a ton of detail, but in case you've forgotten or weren't there, what also makes CellProfiler pretty special is this idea of the image analysis pipeline, the idea that you're going to create a single workflow that's applied to as many images as you have. And that could be one, or that could be over 1 million. And CellProfiler will work exactly the same way, whether you have 1 or 1 million images. And that's what makes it really valuable for screening scale and high content. Each module is then configurable when you're working in the GUI mode so that you can go through and change the settings to customize them to fit all of the principles and practices that I just mentioned. This is the most important button in all of CellProfiler. CellProfiler is designed to be really flexible and customizable. And to do that, it needs a lot of different things in it. CellProfiler, the last time I counted, has something like 80 modules, each of which is a step you might do to an individual image. And 80 is a lot to keep track of, and some of them do a large number of things. So if you're ever lost or not sure what to do, you can use the help button and it will tell you about what that module is designed to do. Each setting also has an individual help button. We are absolutely not the best image analysis tool for every job, but we think we may be one of the best documented and most volumetrically documented of any image analysis tool out there. You can run CellProfiler in 2 modes: in test mode, where you're actually tweaking the settings and playing with them and seeing how they work, as well as in analysis mode, where then your analysis is locked and applied to, again, however many image sets you have, whether that's 1 or whether that's 1,000,000. The modules are broken down into a number of categories depending on what kind of thing you would like to do. Things like file processing, images coming in or files going out, image processing, object processing, measurement. And all of this is searchable, so that if you say want to figure out an object's area, but you don't know exactly which module might do that, you can search. And you will, if the word is in the title of the module, you will actually bring it up in this list of modules that's here on the right. Otherwise, you will find everything where that word is used in the help text. So if I use size, I will get the measure object size and shape module. And if I search area, it won't come up in the module name, but it will come up as part of the module help. Speaking in broad terms about image processing, as you guys probably know, classical image analysis segmentation basically always works off of the principle, my objects of interest should be bright in this image and everything else should be dark. This is how basically all classical computer vision is designed. For segmentation purposes, any transformation you can think of to do this is legal. And this is why CellProfiler contains so many image processing modules. This does not mean that you should measure the images that you might do some pretty radical transformations to. Measure how you treat images that are going to be measured is a whole separate thing that I won't get into today, but there's great resources on this online. But for something that you are just using as part of a segmentation, anything goes. So you may need to enhance dim signal that's really there, or you may need to mask out bright signal that is there but isn't what you care about, like debris, or it's something that is dead cells or other aspects. The more transformations you do before your segmentation, the more critical it is to check those against your raw images. And so CellProfiler gives you several different ways that you can do that. Because it can be very easy to say, oh, my objects are correctly identified for the set of pixels that went into identify primary objects or some other identification module, but you want to make sure that ultimately you're still reflecting the raw image that went in the first place. So we offer a couple of modules that we actually recommend you use in every pipeline, at least one: overlay outlines and overlay objects, to create a record of what you've actually segmented and measured. Trust me when I say that nothing is more sad than going down the route of bioinformatically thinking you found a cool result and only then looking at the segmentations and realizing your segmentation was bad. It's a very sad day when you've done that. But we also offer something that we call the workspace viewer, which was in CellProfiler 2, gone in CellProfiler 3, but is back in CellProfiler 4, which allows you to dynamically, while you're in test mode, overlay multiple channels, multiple objects, and even measurements, so that you can stay focused in on a single difficult to segment region of your image and see how changes you're making to your various module steps affect what you're doing. I'm not going to read all of this, but these are just some things to give you ideas about what are the kinds of image processing steps you can do in CellProfiler to find the things you think are interesting. I do want to particularly call out, though, the run ImageJ macro module, which again is something that was in CellProfiler 2 and is now back in CellProfiler 4. We know that CellProfiler has a tremendous number of things in it, but that ImageJ has even more things. And so we've created a way where you can call your system's copy of ImageJ. And if you're comfortable writing ImageJ macros, can access anything that's in ImageJ by sending that macro into ImageJ and doing your processing on that within a CellProfiler workflow. Once you've found the objects that you care about, they might need some tweaking. So you can do things like making them larger or smaller, editing them, combining two object sets. So if you have bright objects and dim objects, but they're ultimately the same class, you can identify them separately and put them back together. The other thing, in addition to just sort of the reproducibility aspect of the pipeline that we think makes CellProfiler particularly valuable, is this idea of large measurement suites, which again, I'm going to spend the whole second half of the talk hopefully convincing you why that's actually really important. So if you look at the measurement module category, you'll see that we have a large number of measurement modules that you can add to a CellProfiler pipeline. And each one of those actually generates somewhere between a few and sometimes hundreds of measurements. I want to emphasize that many of these metrics can be found in lots of tools, and we are not saying that only CellProfiler can do this, but CellProfiler makes it easy to generate lots of measurements quickly. But getting the measurements is more important than the tool you necessarily do it with. And so you can measure objects or images in a whole different variety of ways. When you add the measure object intensity or measure image intensity modules to CellProfiler, you get not just, say, the mean intensity, but you get things like the integrated, the minimum and maximum, the upper and lower quartiles. You get a whole range of intensity measurements, each of which might be the one particular measurement that is really nice for the thing that you care about. And you can mix and match objects with channels as long as all of your images from the same are taken from the same field of view at the same time, or if they can be aligned in such a way that it makes it be in the same XY coordinates. So I can find nuclei with DAPI and measure the GFP intensity inside of them. Object size and shape is often something that is really valuable. And we offer, when you add the measure object size and shape module, a number of different measurements, and not just area, but things like eccentricity. Is this closer to a circle or closer to a line? Things like the orientation, but also some things that are not actually terribly human intuitable, such as Zernike polynomials. Don't worry too much about the underlying math here, and also please don't ask me questions about it, because I'm going to explain it to you to about the limits of my understanding. But Zernike polynomials essentially measure the similarity between any individual object, and these are just the first few Zernike polynomials, but various abstract mathematical shapes. So they're not really anything that makes sense to the human brain, in the sense of, if I showed you a picture of a cell, you might not be able to easily say how nicely it matches each of these individual shapes. But they're undeniably biologically valuable in terms of giving us accurate descriptions of an object's shape. So here are some objects that were, or images that were taken under a microscope of cells with different antibodies, which have created different shapes. And these are the reconstructions that are created just by measuring the Zernike polynomials and then adding them back up. Each individual polynomial may not be that intuitable and may not be that valuable. And clearly the left and the right aren't exactly identical pictures, but you can generate a really detailed description of shape that's way more than anything that you could get from just something like the perimeter or the area or the orientation. And so that allows you, in a sort of compact mathematical way, to get pretty involved shape description. Something that's a little bit more human intuitable is the intensity distribution of individual objects. So if I have intensity in a particular channel, is it pretty equal from the outside to the inside? And from the outside to the inside, is it pretty much equal between left and right? That might be something that would tell you about the polarity of a molecule that you're interested in, for example. And again, returning to sort of the less human intuitable, things like texture measurements, which again, all have various underlying mathematical definitions that are linked to in the module help, but essentially just come into when I break my image back down into thinking of it as individual pixels with individual value. In a given distance and angle from starting from any one pixel, what can I say about the pixel a certain distance and angle from it? Is it the same? Is it different? Can I easily predict that other pixel from this or not? Is there no information about a pixel a certain distance away based on the pixel I'm centered on? Again, things that aren't necessarily easily human intuitable, but ultimately can give you quite a bit of information. And I'm emphasizing non-human intuitable for something when we talk about later about using measurements that aren't human intuitable at all, such as those that come from deep learning. Once you've created your pipeline and you've added, again, hopefully as many measurements as you practicably can, you can run large image sets on CellProfiler no matter what your personal definition of large image sets is. As was mentioned in my intro, we collaborate with, on average, about 30 different groups a year, and everyone says that their image set is big, and everybody's image set is, of course, of a wildly different size. So if your definition of big is a few hundred images, you can probably run this on your local machine. CellProfiler will automatically what's called multi-thread, and so spin up numbers of identical copies of CellProfiler up to the number of CPUs. So if you have 8 CPUs, which is a pretty standard configuration, CellProfiler can run 8 images at a time instead of 1, making your analysis run 8 times faster. You can also change that number so that if you don't want it running 8 copies at a time, you only want it running 1 or 2. That's what it will do. If your definition of large is a few hundred to a few tens of thousands, if you have access to a cluster, you can talk to your local sysadmin about running on a cluster, either by installing CellProfiler there or by taking advantage of image analysis containers in Docker. And we have instructions on getting started. I'll make sure that this slide deck gets sent to you guys so that you have access to everything that's in the links. And if your idea of large image sets is millions, you can also consider cloud processing. We actually make a program that we call Distributed CellProfiler for running CellProfiler for large image sets on Amazon Web Services. And so we've run something like 100 terabytes of images in the past year on AWS, all with Distributed CellProfiler. I want to now, though, shift the thinking from sort of how you might be thinking in terms of knowing what particular phenotype you want to measure in an experiment to the idea of what if you just sort of let the image measurements dictate the course of your analysis. So talking about looking for your unknown unknowns with an idea that we call morphological profiling. And the idea of morphological profiling really comes down to this idea of when you have a 1,000 by 1,000 camera, you have 1 million digital pixels every time you snap it. And again, a lot of cameras these days are much bigger than 1,000 by 1,000. But this paper is a few years old now, so it's possible that this has changed, but I don't think it's probably changed all that much. Most biology, and even biology that calls itself high content, only uses 1 or 2 measurements that are present within that image, which means you're starting with millions of pixels and pulling out just one metric. And it's fine if that's the metric that you need, and a tremendous amount of good biology, including biology that we do ourselves, is done that way. But you're leaving a tremendous amount of data on the table, and you're sort of incredibly overpowering by taking millions of pixels to only pull out 1 or 2 metrics. And so this idea has been circulating in my boss, Ann Carpenter's brain for a long time, and really wanted to come up with this idea of how can we use the rest of the information in those images. Because there is a tremendous amount of information that's hidden in images. And as I mentioned, or as I alluded to sort of at the beginning of my talk, not all of it is terribly accessible to the human brain. These are a bunch of images of cells in different cell cycle phases. If this were an in-person talk, I usually then ask who would like, from the audience, would like to volunteer or tell me the cell cycle phase of all of these. And I've never gotten anybody to raise their hands on that, not once. You could probably guess that towards the end, these cells are starting to divide and that therefore these might be in cell cycle phase order. But certainly, at least in these first few cell cycle phases, I can't tell you what's the difference between a G1 or a G2 cell, purely from a bright field image. But it turns out computers can, and they can do so with really high accuracy, purely based on measurements of bright field. And even the mistakes that the network makes kind of make sense. So I should say here what's going on is there's also DAPI in this experiment. So the cells are classified into their true cell cycle phase based on things like DAPI. And then the computer is allowed to use only the measurements of the bright field channel to then predict the cell cycle phase. And again, it does this with a really good job, and the mistakes that it makes are things like being somewhat confused between the G2 phase and prophase, which again is not a terribly switch-like boundary. There's definitely more discreteness there. Likewise, with S phase, it can be a little bit confused with G1 and G2, but again, though it is not a perfectly switch-like biological state, there are things that happen along the way. And so at least some of that error is due to our sort of human need to categorize everything into discrete categories, even though biology may not actually be as perfectly discrete as we would hope. And this is just a pretty spinning movie of saying, if you look at the bright field features of each of these cell cycle phases, you see that the ones in the same cell cycle phase all cluster together in this pretty spinny projection movie. So with this idea that there's hidden information in images and that the pixel information, the quantitative information is there, what kinds of new experiments can we imagine that take advantage of that extra information? So again, this sort of birthed the field of morphological profiling, which again is not purely out of the Carpenter lab. And there's a number of labs who work in this field, but she was certainly one of the people involved near the beginning. And the idea is just to take genetic or chemical perturbations, to do experiments. It says here in multi-well plates, but you don't actually need a tremendous number of samples to do this. We've done this in a single plate, in a single 96-well plate with only a couple dozen different samples, and sometimes even smaller for things like CRISPR clones. You don't need a large number of samples to do this. All you need is a large number of measurements. So you put this on an automated imager, whatever kind you happen to have. You do image analysis, whatever kind you happen to want to do. And the real magic in the technique is, again, just creating these large measurement sets that have as many measurements as you can possibly manage to fit. Because you can then see how individual measurements sort of go up together or go down together and associate in ways that allow you to group the perturbations in your experiment in sometimes really surprising ways. So one of the first techniques that actually proved that this would work was an experiment that our lab did, where they took drugs that had a known mechanism of action class, and they put them on some cells that were stained with three what we think of as relatively simple dyes, a nuclear dye, an actin dye, and a tubulin dye. And they said, can we actually then group the drugs back into their known classes, say that all of the drugs of class A belong together purely from actin, DNA, and tubulin. And it turns out that you can. It turns out the nucleus alone is actually enough to give you 67% accuracy, and pretty good accuracy even for some drugs that are not, you don't think of as terribly nuclear, things like actin disruptors. You can do with pretty decent accuracy, again, just from looking at the nucleus alone. If you include just the nucleus, actin, and tubulin, you get 94% accuracy of being able to say, I know which of these drugs belong together. And again, the mistakes that we're seeing are things that make sense for the most part. They're things like DNA damage being confused with things that change DNA replication, and those two processes are incredibly interlinked. So again, the confusion is as much about our need to biologically give everything one category as it is about anything that was actually wrong in the algorithm. And so this was one of the first things that sort of proved yes, you can actually do this in an unbiased way and learn things about the treatments in your experiment purely from dyes that are not targeted to your favorite gene, but just sort of showing you where the structures of the cells are. This is not work done by our lab, but is work that is created what is a company called Recursion Pharmaceuticals, which my boss is a board member at. And one of the other sort of early proofs that this idea of morphological profiling could really work. So they were studying cerebral cavernous malformation, which they could induce with an siRNA. And the healthy cells are on the left and the diseased cells are on the right. And there's a really striking visual phenotype here. Anybody who spends a lot of time looking at cells would say, oh yes, I can definitely tell the cells on the left from the cells on the right. So what they did was actually something pretty smart, is they did a drug screen here, and then they after taking all of the pictures of all of the drugs that they tried, they gave the pictures to human experts and they put them through a high content imaging morphological profiling approach also. And they said to the human experts, pick out the best few drugs that you can that make the siRNA treated cells look most like healthy. And they did the same with the computer. And then they sort of pitted the humans against the computers by giving both top hit lists to mice and seeing how the mice did. And it turns out that the human experts did not actually pick out the right features of what a healthy cell really looks like. And their drugs did more poorly than the drugs that were picked up by the computer as creating the most healthy-like phenotype. The computer was able to not have the human bias and actually focus on what a healthy cell truly looks like at a pixel level and suggest better drugs to treat a particular disease. So again, the fact that these approaches succeeded was one of the things that led our group, along with Stuart Schreiber's group, to create what we call the cell painting assay. And the idea for the cell painting assay was just, rather than studying then one particular gene for one particular disease, let's stain as many parts of the cell as we can, as cheaply as we can, with reagents that are as easy to get as we can. The list of dyes is on the slide here, but most of them are things you probably have in your freezer already, things like Hoechst and Floyden and MitaTracker. So that we can create a really, really dense stain panel so we can generate lots and lots of measurements. And so we have a slightly updated version of this protocol, which should be out hopefully in the next couple months on bioRxiv. The protocol is actually on our website now. But by doing this, we're now able for each cell in the cell painting assay to generate about 6,000 measurements of every individual cell in our experiments. So what can you learn when you start doing this really dense measurement of cells? So this work was led by Mohammed Roban, who was a PI in Ann's lab and now has his own lab, looking at cases where we took a cancer cell line and overexpressed a bunch of individual genes, either wild type or mutant versions, to see could we group the genes into their biological pathways based purely on this cell painting experiment. And so zooming in a little bit, here you can see a bunch of members of the RAF and RAS pathway that are all being associated together. And right next to them are a couple of BRAF and RAF mutations, constitutively activating mutations specifically. And so by cell painting, which again has no particular RAF or RAS marker in it, we could see that these activating mutations are similar to overexpression of the wild types, but not completely similar. They have their own distinct phenotype. And again, these are mutations that are known to cause disease. And so when we first did this experiment, Mohammed looked through all of the pathway, not just could we sort genes into pathways, which it turned out we could, but what were the interactions between pathways that we could find? And we found a lot of stuff which was known in the literature, which is comforting. You would like to know that you can rediscover known things. But he also found at least one pathway interaction between the NF-kappa B pathway and the Hippo pathway that nobody we talked to could find any evidence for and didn't have any reason to believe was real. So we're a totally computational group, but we convinced somebody to build us a reporter assay. And it turns out that when you make a reporter assay for the Hippo pathway and you overexpress NF-kappa B pathway genes, it actually turns the reporter way down. So we're actually able to detect a anti-correlation between two different pathways in a cell just by saying that their cell painting signatures looked opposite to one another. And so this technique really sort of proved to us that yes, looking just at cell compartments with cell painting allows you to actually get really specifically into the details of biology. And so this is some unpublished work that's currently being led by Marzia Hagigi in our group, looking at skin fibroblasts from patients with psychiatric disorders. And what she showed is that even looking in skin fibroblasts, again, we're not in the right cell type at all here. We're able to see some differences between control patients and patients with different psychiatric disorders, and different disorders group differently here. And just to emphasize to you one more time how bad human brains are at this, this is the feature that she's found is most important in separating those some of the really different patient groups. And I've looked at these images a very long time, and I can't tell the difference between the images on the left and the images on the right. That information is not available to the human brain, but it is available to the computer, which allows us to hopefully try and imagine things like doing drug screening in a skin sample for people with psychiatric disorders. And so in the last 10 minutes or so here, I want to talk about how we can push this further. What are some things that we can do to try and do even more of this? So one thing is to just, I mentioned that there were a lot of rules about trying to find objects, and that trying to find objects was sort of 90% of the battle. So that's an obvious area to try to improve things to make it a little bit easier to do. So one thing we did a few years ago was create a Catgill challenge for what we called the universal nucleus detector. So we had images that had a wide variety of visual phenotypes for what a nucleus was. Our lab basically stopped doing everything else for a month, and 12 people hand annotated 28,000 nuclei. And we asked people to create a network that could find a nucleus no matter what the background looked like. And the CellProfiler reference is shown here, and you can see it does pretty well. It has access to the test data, so it should do pretty well, but it doesn't do as well as some of the networks that don't have access to the test data. And so the top performing model actually does better without any extra configuration time than a CellProfiler expert does, with time spent configuring individual pipelines for these individual image categories. And so we think that deep learning can potentially be really powerful in object finding. And so we've released all of these hand annotated nuclei publicly. And one of the first tools that we saw that came from that data is a tool called Nucleizer, which allows you to upload a picture and download a map of your nuclei. But that same training data that we generated has also been used for some tools that you might have heard of that have come out in the last couple of years, such as Cellpose and StarDist. Both include that data science bowl training data as part of their training set. And recently we've added CellProfiler plugins to be able to install both of those from source to make the hard part of image analysis, object finding, a lot easier. So can we also find better features? I mentioned that we're right now with cell painting getting about 6,000 features per cell, and that's pretty good. But could we do even better? Could we find even better features than the 6,000 things that we're currently measuring? So Juan Caicedo, who was a postdoc in Ann's group and now runs his own lab at the Broad, was really the person who led the beginnings of this work in the group. And what he said is essentially, deep learning feature measurements, and I'll talk a little bit more about exactly what that means, are just as valid to describe a biological image as things like a Zernike feature or a texture or even an intensity. And a lot of those human brains are really bad at understanding what they mean anyway. So if we don't care about necessarily understanding what the individual biological features in our images are, we just know that we need a lot of them, can we use deep learning, because those are numerical measurements of images and objects that are just as valid as something like a texture or a Zernike feature. And if you've heard deep learning a lot and never actually dug into what it means, this is my 2 slides that will tell you about sort of the very basic ideas of what deep learning is. I will warn you, this website is very addictive. You can spend a lot of time watching neural networks train in the browser, but a neural network is essentially, in the context of biological images, just a bunch of image filters that are passed over an individual image. And depending on how much each filter is fed into the next one, you can create essentially different abilities to predict. So this is a before training neural network, and this is an after training neural network. And you can see here the lines that connect each filter in each layer to the filters in the next layer are now different widths. We call these filters, and we call these weights. And the idea is just that by sort of strengthening or weakening the connections between all of these, we can actually make some pretty valid models that understand what's going on. This is clearly not a perfect spiral, but it's not an imperfect one either. It definitely sort of is starting to get the idea of a spiral. And so by taking what these filters look like and the mathematical numerical values of these weights, we can treat those exactly the same way we treat a texture feature or a Zernike feature and use it for morphological profiling prediction. So the experiment that Juan did is an experiment that sounds kind of silly when you first talk about it, but the point is not to actually get a network that works, but just get a network that understands the task. And it was for pictures of cells to guess which drug they were treated with. I say this is a silly experiment because we're going to almost always know what drug a cell was treated with because we're the ones doing the treating in the first place. We want to just stick a drug on a cell. But by training a network where the network thought that that was the goal, to guess which drug a cell was treated with, Juan was able to make sure that the network learned the idea of how cells change in response to drug treatment. And so then when he took the network that came out of that and took the feature layers and the weights, essentially took all of those connections that the network had learned, that same analysis with the drug treatment, I said, with nuclei, actin, and tubulin could get 94% accurate, grouping drugs into treatment groups. One could get it 97% accurate, and he could do it 700 times faster. And so these deep learning based features, even if they're not human understandable, clearly do a good job of describing what a cell looks like and can be used to group cells into categories. And so he's working on something that we call Deep Profiler to make this easy for everyone to use. And in just the last few minutes, I'm going to tantalize you, hopefully, with some ideas of some new ideas of experiments we can imagine coming from this. One is just, as I said before, the magic in this technique is in the lots and lots of measurements. It's not in anything to do with the particular dyes that we're using. And so we're starting to work with some groups to create customized dye panels for particular biology, in this case, lipid biology and adipocyte maturation biology, and take this same morphological profiling approach, but in new stain panels. We know that there's information hidden in bright field. I opened the section with that. And other groups have worked on how can we predict where organelles are from bright field images alone. By taking in exactly the same way as the first cell I showed you, or the first slide in the section that I showed you, where you have a nuclear image and a bright field image, and you try and predict one from the other, it turns out that you can make some pretty reasonable predictions of what a nucleus would have looked like in DAPI based purely on the bright field. And so for different organelles, this works to different abilities, but you could imagine somebody adding bright field measurements to an experiment and adding stain prediction in so that you could have more theoretical channels. One thing we're also working to do is to be able to use the cell painting data to hopefully replace some other experiments that can be harder and more difficult to do. This is work that was sort of taken over the finish line by Greg Way, who is a postdoc in Ann's lab, who starts his own lab next week. where we took an assay that was designed to look for particular phenotypes, cell cycle phase, and some measurements of viability, things like cell depth and particular apoptotic markers, and took some cells that were treated with various CRISPRs, split the plates in half, did these cell health assays on half of the plates and cell painting on the other half, and said, how many of these different phenotypes that we were predicting by using specific stains could we learn to predict just from cell painting stains? So can we replace these with a cell painting assay? And so it turns out that for a lot of these, you can, with pretty reasonable accuracy, predict what the cell health assay would have looked like purely from the cell painting data. Shape measurements are overrepresented at the top here, which isn't necessarily super surprising, but even some things like apoptosis and certain kinds of cell death are visible in cell painting, even though that is not actually what is particularly being stained. And so we're working to create a really large cell painting data set, what we think is going to be the world's largest publicly available cell painting data set, as part of a consortium with 10 pharma companies, 3 nonprofits, and ourselves. This should have more than 100,000 compounds and at least 15,000 genetic perturbations, with the idea that anybody can then, once this data set is released to the public, try to figure out what's going on in your biology by comparing it to cell painting of the same drugs or of the same mutations, and be able to do essentially this experiment of not having to do every experiment, since cell painting is relatively cheap and easy. If you can learn the results of a really hard to do assay from a cheap assay, maybe one we've already done for you, you can really start scaling up your biology to really cool levels. In the interest of time, I'm going to skip that one section. But so hopefully what you're thinking at this point is, how can I learn how to do this, and where can I go for help in learning to do this? For morphological profiling in particular, one of the best places to start is a review that our lab wrote with a lot of other labs collaboratively 4 years ago, which walks through the individual steps of how to do this and some recommendations for what are best practices. Ultimately, this paper came out of what eventually hackathon that eventually turned into something that we call the Cytodata Society, which is a society that works of profiling labs that all work together to come up with best practices and to try and advance each other's research. And their annual meeting is next month, so I recommend checking it out. For image analysis in general, COBA is a center that is joint between our team, the CellProfiler team, and the University of Wisconsin folks who maintain ImageJ. And our goal here is to create a center where anybody can go for help with bioimage analysis, and to do this by funding development of new technologies, but also with the maintenance of existing ones like CellProfiler and ImageJ, and particularly with community engagement. The best tool in the world isn't helpful if no one knows how to use it. And so one of the things that COBA does is sponsors the image.sc forum, which is the single best place to get help on any image analysis problem, because it's staffed by experts from, I think the last time I counted was something like 40 different image analysis tools and societies that all use this as a central help forum. You can also look for conferences and jobs and things like that, but you can also just post a picture and say, here's the picture I have, here's what's, I want to know what software is the right software to use on it, help. And you have experts from 40 tools who are going to help you out. And so with that, I want to thank the people in my lab and in Anne's lab, as well as the people who paid for this, and our biology collaborators who contributed all these images. Like I said, we're a computational group. And thank you guys for your attention.
Thank you so much, Beth. That was a good tour de force of everything that one can do with image analysis. If anybody has any questions, feel free to unmute yourself and ask the question, or feel free to type it in the chat. This is supposed to be a very interactive session, especially with cell painting. That's really eye-opening. I'll let anybody else ask the question before I ask any. All right, so while somebody has any questions up in their mind, I'll probably go ahead and start with the first question, which is cell painting, of course, right? One of the ethos of your lab, as well as Ann Carpenter's lab, is make measurement first and then ask questions.
Mhm.
With Juan's paper that you mentioned, it's very easy to go to the stage of getting all the measurements with CellProfiler. The biggest hurdle is how to apply various bioinformatics pipeline to understand the relationship between the measurements. So what would you suggest and recommend for that step?
So in terms of understanding the interactions between the features, there's definitely a lot that you can do in terms of comparing how individual features look against one another. And in fact, we don't actually typically keep all 6,000 features. We try and remove ones that are sort of really heavily correlated with one another and get it down to what we think is the most unique, 500 to 1,000. But yeah, in terms of then being able to then compare samples to other samples, we have a handbook online with how to take CellProfiler features and do exactly these sorts of experiments. And again, this paper, you don't want to do it exactly the way our lab does. If you want to see the best practices of the whole community, this Caicedo et al. Nature Methods 2017 is really an awesome place to get started.
No, that's great. Thank you so much. If you have any questions general about CellProfiler 2, please do Dr. Cimini know about that. I might have done a very terrible job last week on that, and she can really do something much better than I can.
I'm sure it was not terrible, but yes, happy to answer CellProfiler questions too.
So Beth, this is fantastic talk. This is Jenny Yang from Georgia State University.
Hi.
Hi. Certainly, I need to catch up reading for your publications. So I wondered, have you using CellProfiler to look for the subcellular feature changes? Like VR and a Golgi and things like that?
Yeah, so we often don't, that's a great question. So we often don't make direct measurements of things like the ER and the Golgi in terms of, we don't call them objects, but we absolutely do measure them. And that's in fact, a big part of those 6,000 measurements that I mentioned. We have both an ER stain and a Golgi stain in our cell painting panel. And we use things like the texture measurements, the intensity distribution measurements, and also some other ones I didn't talk about, like granularity, to look at those in particular. It's an open question as to whether or not, if we identified them as objects, we'd learn even more. But it's just not a question that we've had time to dig into yet.
So I want to follow up on the texture measurement part. Typically, the intensities depend on the bit depth of the image. However, CellProfiler ends up scaling those intensities from 0 to 1. And when you were mentioning about texture, you said that there is a particular pixel, which will be the reference pixel, and you want to see how the intensity varies from that reference pixel to the pixels on the other directions. So does that really matter that the scaling has been done or not? And if I want to think about this in a particular example, let's say you want to study an organelle such as lysosome, and with regards to, or endosomes and lysosomes, I should say over here, if there is some kind of a perturbation, if they are getting enlarged versus getting tiny, would the texture feature really be able to detect that?
Yeah, so the scaling 0 to 1 is in fact most of the things that I'm aware of that do calculation of texture features in this way start by actually scaling the image 0 to 1, and by in fact usually going further and scaling just the image into a number of discrete bins. So the original paper actually just said we're going to take the image and bin it into 8 bins. And so it doesn't really matter that the things are scaled 0 to 1, because the relative difference when you're talking about 0 to 1 or 1 to 65535, the relative differences are always going to be the same if you've done your scaling properly. And yeah, texture and granularity measurements do help us find the differences in sort of like small organelles, like do they get bigger or smaller? There's a paper that we've done with another group that is going to be submitted soon, where they've looked at things like it's how the granularity scale, which I didn't talk about granularity, but granularity essentially is a measurement where you say, all right, I'm going to delete all of the dots of size 1 from the image, how much signal is left. Now I'm going to delete all the dots of size 2 and 3 and 4 and so on and so forth. And so that measurement, even though you're not identifying things as objects, gives you a really, really sort of good measurement of how many lysosomes or endosomes or whatever of sizes 2, 3, 4, et cetera, are present in your image. And we're able to get things like or suborganelle size from those kinds of measurements.
Well, that's fantastic. So if anybody wants to, typically you have this awesome tutorials or step-by-step of workflows per se. If somebody wants to do an entire workflow of analysis, is there a data set and associated workflow available that one can just follow and walk through with that?
Yeah, so on our cellprofiler.org website, we link to a number of tutorials and examples that we have. One of our tutorials we call the advanced segmentation tutorial, which is actually uses some of the data from this particular, this PLOS One Gustafsdotter paper, and has a cell painting like, it's actually more complicated than a typical cell painting analysis because it does actually go in and find some of the organelles, but gives you an idea of how to build such a pipeline, and also how to handle things like debris and blurriness that are going to come across in real world images.
Well, that's fantastic. Anybody else has any further question? Go ahead.
Yeah, so my lab has developed the, genetically encoded, calcium sensor for really report about subcellular calcium dynamics. So have you applied, for your methodology to follow the calcium signaling?
Yeah, we've definitely worked with some teams. We've never done it in the context of cell painting, but certainly we've created cell-profiler workflows to look at calcium signaling. I know we've done some publicly on the forum. There's some also in publications. If you go to our list of publications, there's definitely some there.
Okay, all right, thank you.
Hi, so in your drug discovery program, do you compare your data based on whether cells are looking healthy or not, or are there any measurements that you go for? And another thing is in CellProfiler, is there an inbuilt module to compare the cells whether they are healthy or, how different are they?
Yeah, so healthy and not healthy is a really interesting question. And again, we sort of get at it in that cell health experiment that I described. There's a lot of different ways for cells to be unhealthy. So we don't typically sort of come at it from the point of view of we want to look for healthiness of cells, but where that does end up tending to creep in is in drug screens that we do. If we end up seeing that a lot of drugs that at lower dose points don't look all that similar, at higher dose points all start looking pretty similar, we're like, oh, well, those just must be making the cells sick and sad. We don't have a way to in CellProfiler, so with CellProfiler, we typically are not usually making sort of classifications. There is a classify objects module, and there are ways to sort of do classification in CellProfiler, but typically we think of CellProfiler as sort of making the measurements and then the sort of assigning into phenotypes we almost always do downstream. But there are ways, if you use tools like CellProfiler Analysis, to do that sort of a classification, to bring that back to CellProfiler and keep applying it to future data sets. But healthy is going to look a little bit different in every situation, and so you'll have to train your own for your cells.
Thank you.
Okay, so we're on top of the hour. If somebody has one last burning question, please go for it. I don't want to hold Beth here for a long time. If not, I guess I want to thank Beth once again. Thank you so much. Really appreciate your time, Beth, and clap from everybody over here. I really look forward in future for the jump cell painting database that will come out, and the deep profiler, which sounds very exciting. I guess the whole deep word is kind of dumbing down everything that one has to think about. Press one button and boom, everything comes out on the other side. So that's going to be exciting. I hope you are okay with some people reaching out to you. I mean, she's very easy to find. You can type her name and Broad, and her email should pop out. So Thanks again, Beth. Thank you again so much.
Yeah, thank you so much for having me. It was great to talk to everybody, and I hope everyone has an awesome rest of their day.
You as well. Bye.
Bye.